All right, so I'll start in on the introduction. And so I think many people here may have the pleasure of already knowing Shahab, but Dr. Shahab Vahdat, he's an assistant professor in APK, Applied Physiology and Kinesiology. He has joint appointments in the Department of Biomedical Engineering, has a long track record of training, having started off with a Bachelor's of Science in Electrical Engineering from the University of Tehran, and a Master's of Science in Biomedical Engineering from Sharif University of Technology, PhD from McGill University in Movement Neuroscience, and then postdoctoral training in neuroscience and brain imaging at the University of Montreal, and further work in neurology and brain imaging at Stanford. And so as we were just talking about, Shahab got a Brain Center Neural Injury Award in 2022 and was actually on a topic that he'll be talking about today. And that work actually was able to propel him to getting an R01 in 2023. So that's exactly the way we like to see the center seed grants and funds kind of work, is to kind of help give people a chance to kind of take that next step and provide that support. And as Jessica pointed out, he's even since gotten another R01. So Dr. Vahdat's research focuses on neuroplastic mechanisms involved in stroke recovery and motor learning. His lab uses advanced multimodal optogenetics and neuroimaging technologies to study and modulate plasticity at the molecular, neuronal, and systems level. He's made significant contributions to understanding brain circuit reorganization following stroke with neuroplasticity and motor and sensory learning, spinal cord functional imaging, and motor memory consolidation. He is well published, with his work being published in top journals including Neuron, Journal of Neuroscience, Stroke, and PLOS Biology. And as Shahab reminded me, sometimes the best writing he does, he and I share a writing space. It's been very effective, I think, for both of us. Other people call it Starbucks, but we call it our writing space. But without any further ado, Dr. Vahdat.
Thank you very much, Michael, for the invitation and for the nice introduction as well. So it's great to have this presentation here. As you mentioned, I received the seed fund from the brain, and it really helped us to collect the pilot data and to get things started, and then get the following NIH grants. So today I like to give some overview of the work that we have been doing in the last 4 or 5 years at UF. And of course, some of these data are preliminary because the R1 we received last year, and it takes some time to start data collection. But we will have some data patient, like from 3-4 patients per group. At the end, I will also provide some update on that. So I'm at the Department of Applied Physiology at UF, and my lab, Sensory Motor Plasticity Lab. So I have a small kind of lab with few PhD students and some clinical coordinator research staff. So we have 2 streams. We study both in human stroke patients and mouse models of a stroke. The reason we use the mouse models is we can do more causal intervention to see the effect of our treatment. So in human, currently we mainly try to characterize the pattern of changes following a stroke, with the hope then to identify targets for potential targets for neurostimulation, trying to shape the circuits following the stroke, and in this case, move away from maladaptive plasticity. So that's basically the main goal of my lab. So today, I will mainly focus my talk on the main motor impairment that we are now focusing in the lab, spasticity, which I described is one of the main impairments after the stroke. I'll give an introduction, what we know about the spasticity. Then in the second part, I will briefly talk about kind of the methodological development in my lab, trying to characterize the function of the spinal cord and brainstem networks. You will see why this is important for the studying of spasticity. Then I will provide some data that is now submitted we have 3 papers that are submitted now. This is a good time, I think, for the lab. That is more a characterization of them using this method developed in the healthy participant, in the healthy control. We look at the characterization of the descending and the spinal brain stem networks. And in the last part, as I mentioned, I will update how this can be applied in our stroke population to study spasticity. So, as you all know, this is a small group. So stroke is the sudden interruption of the blood flow, and then it will cause the neuronal loss. And this is the leading cause of this long-term serious disability. And unfortunately, the recovery of upper motor function and hand motor function is often poor. And there are not much neuro-rehab treatment. There only exists two FDA-approved treatment for a stroke, RTPA and thrombectomy, which both they can be only administered at the acute stage, only a few hours after the stroke they will be effective. And for the course of the chronic stage, which is weeks to months, is usually 6 months after the stroke in humans, there is no known treatment, actual no treatment for a stroke. And there is definitely critical need to invest in this. And why this is important, because I like to emphasize that after the stroke, there is this immediate neuronal loss and degeneration that is just within the minutes to hours after the stroke. And this process is, of course, irreversible. So we will end up with a lesion core. But within days to months after the stroke, weeks to months after stroke, there is this secondary degeneration and this kind of gradual pattern of neuronal damage that will follow on. And usually people, they assume that this is just in the area surrounding the lesion core. But now we understand that this also happens in other areas of the brain, even the remote areas far from the lesion core. And this process of this maladaptive plasticity, or sometimes this neuronal loss, the secondary damage, is reversible. So, and this is happening during this chronic stage after stroke. So basically the goal of my lab is to try to understand these maladaptive kind of processes after the stroke, and trying to see if we can have some sort of treatment using neurostimulation. And the area that I'm mainly interested in my lab is the spinal cord. So as it turns out, spinal cord also undergo changes following a lesion of the brain, which is not very surprising considering all of the descending pathway from the brain to the spinal cord. So from the animal studies, we know that there is a huge rewiring of the spinal pathway after the stroke. There are rewiring between the two hemichords in the spinal cord. There is axonal sprouting related to different descending pathway as well. From the animal model, from rodent studies, we know that there is inflammatory response, like factors like TNF alpha, IL-6. It will increase in the spinal cord locally after a lesion of the brain. Also, there is increased concentration of the neurotrophic factors. That both of these, they also correlate with the amount of recovery after the stroke. So it seems that these early changes in the spinal cord is important for the pattern of recovery following the lesion of the brain. And not only that, we recently know that the spinal cord is not just a hub just to convey the motor cortex commands to the muscles. There are these local circuits in the spinal cord that are important and involved in motor control, as well as in the brainstem. So, for example, in the case of a cortical stroke, these areas might provide new targets, trying to, again, use this plasticity and move it away from the maladaptive side. Two major impairments after stroke that we know that they already have a spinal cord components are: one is the spasticity, that is going to be mainly the focus of today's talk, and the other one is the weakness, force control deficits. We know that is also related to degeneration of the corticospinal pathway. It kind of correlates with how much this pathway to this spinal cord has been degraded and damaged. So these are like some studies that I did when I was in McGill University. We quantified the corticospinal pathway using the DTI imaging, diffusion imaging, and it correlates very well with the, as not surprisingly, with the amount of weakness, force deficits that these stroke patients they show. But today, as I mentioned, I'm going to focus mainly on the spasticity. The definition of spasticity is this involuntary contraction, which is their characteristics is that they are velocity dependent. It means that if you move the joint faster, you will feel more resistant. And in the upper limb, they are mostly on the flexor muscle side. In the lower limb, they are on the extensor side. And spasticity, as you can imagine, because it's velocity dependent, it has something to do with the reflexes, with the spinal reflexes, which they use this sensory motor pathway. And in a spastic patient, we have usually this hyper excitability of the stretch reflex, the spinal reflex, which causes this increased muscle tone. So we have two symptoms here: hypertonia, which is this velocity dependent muscle tone, which is different from rigidity, which is not velocity dependent, if you want to separate, distinguish between them. And also we will have hyperreflexia. So both of these usually occurs in the same patient, and we don't understand quite which one leads to the other, or both of them, they correlate to each other, basically. And in terms of its management, spasticity usually first of all, it's a delayed process. Usually it occurs in some patients 1 month after the stroke, so they don't show early signs of spasticity, but they will develop it later. This is suggestive of that this spasticity is related to this maladaptive type of plasticity that occurs after the stroke. So some brain areas, they balance of excitatory, inhibitory activation changes, and which is not necessarily the best way that it changes, and it causes this additional problem, this spasticity problem for these patients. In terms of the clinical management, usually there are basic treatment for these patients, mostly at the peripheral level. So there are like treatment for the occupational physical therapy, trying to relax this muscle, using Botox injection, pharmacology, again, trying to kind of relax the affected muscle, and or trying some sorts of surgery or stimulation, again, at the peripheral side. And all of these, the main kind of the goal is trying to relax the contracted muscles so that the patients, they can engage in the training again and physical therapy, and they can improve on their motor tasks. But none of these treatments currently, they look at this original path impairment. So this kind of maladaptive plasticity that is being built up over the months is in some cases. And the main reason is that because we don't quite understand how the spasticity develop after stroke. So if you understand the underlying mechanism, we may be able to provide them kind of treatment that trying to divert from the main cause of the spasticity, as opposed to, looking at these peripheral symptoms and effects. And what we know, again, from the spinal cord is that spasticity is correlated with the hyperexcitability of this spinal pathway. Usually in humans, this is measured through the H reflex, Hoffman reflex. When we do electrical stimulation of a peripheral nerve and we look at the EEG response related to this monosynaptic pathway, there is this some famous study from the CATS lab from Paris that they show, for example, for this spastic patient after the stroke, these clinical measurements of the spasticity, which is this usually we use Ashworth scale or modified Ashworth scale in the clinic. It correlates with the degree of this hyperexcitability of the spinal pathway as measured by this H reflex. So both in the upper limb, in the FCR muscle, and in the lower, in the soleus muscle, this correlates very well with the degree of spasticity. And just to mention, this is a specific mechanism in the spinal cord. This is using the conditioned way of H reflex that we do at high rate stimulation, low rate, and we try to look at this post-activation mechanism called post-activation depression, which is related kind of to the depletion of the neurotransmitter in the sensory motor, in the monosynaptic pathway between the 1A afferent and the alpha motor neuron. the degree of neurotransmitter depletion as measured by this mechanism is related to spasticity. Again, this is some local mechanism in the spinal cord related to the hyperexcitability of H, and this is related to the amount of spasticity. But the main question is how the brain lesion at the cortical level or subcortical level, it will cause these changes at the spinal level, these the local changes in the sensory motor synapse at the spinal cord. So we have some, again, evidence from the animal models. We know that at the spinal cord, there's a sprouting of these 1A sensory afferent to the motor neurons. So these neurons, sensory neurons, they will become more extended after this in these spastic animals. And this extension of the connections between the 1A and motor neuron is related to the hyperreflexia in these animals after the stroke. So this is, again, work done in mouse. From the clinical work, we know that they measure, like using, again, some electrophysiological approaches. And some the other hypothesis is that it might be related to the disruption of the inputs to the motor neuron pool, but not just some local processes related to the sensory motor connection. In this case, they measure these persistent inward currents or the PICs, and they show that after the stroke, in spastic patients, it relates to the amount of spasticity. So these are this increased kind of local, again, synaptic mechanism that is related to this plateau potential in the motor neurons, and they kind of cause overexcitability of the motor neuron pool in the ventral cord of the spinal cord and cause this spastic symptom. However, as you know, spasticity not only occurs after the stroke, but after the spinal cord injury, other diseases like ALS or MS. And this mechanism has been shown to be mainly involved in the other source of spasticity after the spinal cord injury or ALS. After a stroke, there has been some studies done, and they didn't find any correlation between these increased PICs and post-stroke spasticity. So it seems that if there is any increase in excitability of motor neuron, which causing this increased contraction, it should be related to the descending pathway weight to the motor neuron pool. So there are two paths that they can modulate the motor neuron excitability. One is the ionotropic, glutamatergic neuronal connections, projection. The other one is this brainstem modulatory drive through the monoaminergic connections, through the serotonergic or norepinephrine. So one of the main hypotheses that we know also from the primate model, from the monkeys model, is that the spasticity in monkeys after the stroke, after lesion, occurs when there is a lesion in the premotor cortex mainly. So this is a very early work from 70 years ago, 80 years ago, that they showed that the lesions of M1 mainly is the weakness, these kind of negative symptoms of the stroke, and lesions of the premotor cortex will cause the positive symptoms of this spasticity, this increased contraction after the stroke. And the other sort of evidence comes from another set of study looking at the brainstem level and doing some lesions in the reticulospinal tract, in the reticulospinal tract, in the reticular formation, medullary reticular formation, that they show that the lesion in this area will cause spasticity. And it's not, these two are related to each other because premotor cortex is the main projection to this reticular formation area. And this reticular formation, again, is one of the descending pathway, glutamatergic pathway to the motor neuron. So this is kind of putting everything together. So it seems that, so this is the current model that we have in humans, that there is a stroke, for example, a cortical stroke involving the premotor cortex as well. It has some projection to the brainstem, including the reticular formation, the medullary or pontine reticular formation, and then they have some projection to the spinal cord. And these projections, some are excitatory, some are inhibitory. Based on the current models, the balance between the excitatory inhibitory projections from the brainstem to the spinal cord is disturbed. So there is now more excitatory connection or projection to the flexor muscles, for example, after these lesions in the brain. However, this is just a model, to be honest, and we don't kind of quite test this model. So this is just a model based on the data that I just showed you, and some of them are very old, from 70, 80 years ago. In human, the problem is that there is no direct way to measure these connections. So it's very difficult to measure the connection related to the descending pathway. The reason is that, the best technique currently to measure brain activity, fMRI, functional magnetic resonance images, mainly focus on recording the activities of the brain. So we cannot quite understand how these descending pathway operate and how this changes in healthy controls or in a stroke patient. And again, there are these electrophysiological methods using H-reflex, but they are very limited in terms of their connections to the brain. Usually they operate locally at the level of the brain spinal cord. So that was one of the main goals of my lab, was to develop a neuroimaging method to be able to characterize these pathways. So between the brainstem, spinal cord, and the cortex. And then it will allow us to look at these connectivity, looking at these hypotheses that we have from the animal model, and to actually test it in the stroke patient. So the second part of the talk, I'm going to go very quickly over this because this more technical part of the work that I've done in the lab is basically trying to develop the simultaneous spinal cord brain fMRI approach that the work had been started from where I was a postdoc, at the University of Montreal, and has been continued in my lab as well. So basically what we do is that we have developed these innovative technologies, these sequences in the scanner to functionally measure the brain activation as well as the cervical cord activation, spinal cord activation. And when we do that, we can then look at the connectivity, what's it called, these correlations between the signals of the brain and the spinal cord using these functional connectivity methods. And this will allow us to characterize this descending pathway, brainstem activation, spinal cord activation during task performance in subjects. Okay, so this is just to show you the functional images that we can get. This is the spinal cord image that we can get, nowadays again, this is 10, 15 years in progress and improving these sequences. We can even see this gray matter, the butterfly. So we have enough resolution to kind of distinguish between the ventral horn, between the dorsal horn, between the left and right side of the spinal cord. And the first studies that we did about this study was started 10 years ago. We first showed that during a motor task in healthy subject, we can detect activation. For example, this is during a hand finger tapping task. So we expect to see activation at around C7 cervical level and on the ipsilateral side. So again, this is one of our earliest protocols, so we didn't have much resolution here to distinguish between the ventral dorsal horn, but we could see laterality in this study. So not only we can look at the activation in the contralateral cortex, in the contralateral, for example, striatum, ipsilateral cerebellum, but we can also look at the activation in the ipsilateral spinal cord simultaneously within the same imaging session while the subject performs this motor task. And this allows us to look at the connectivity now, looking at the correlation between the signals of the spinal cord and the signals of the brain. And for example, in this study, we could look at these task-based connectivity changes. We had a motor learning test, and we saw that the connectivity of the spinal cord to the cerebellum increases as the subject learned, and its connectivity decreased with the motor cortex, with the contralateral motor cortex. Again, this is just providing as an example how this new protocol can be used to characterize task activation in the spinal cord and its connectivity with different areas of the brain. Later on, this is about a study that was published 4 or 5 years ago. We look at using the similar sequences during the resting state period. So this is when the subject, they don't do any task, they lie in the scanner, and we can look at their kind of correlation between different brain areas, in this case also spinal cord, and how they are organized. So what we can do is we can define some regions of interest, for example, in one side of the spinal cord, in the left side and the right side, and we can see all of the brain areas that at rest, their signal is correlated with these regions of interest. As you can see, what we found that during the rest, the left side of the spinal cord, in the cervical cord, it was mostly correlated with the contralateral cortex, with the sensory motor areas, as well as the thalamus, striatum, and the cerebellum. On the other hand, the right side was mostly correlated with the, again, contralateral side, with the left side of the cortex, which is consistent with this decausation of the sensory motor pathway at the level of the brain stem. So we have this dual disassociation. So the left ROI spinal cord is more correlated with the right side of the cortex and vice versa. So this was reassuring for us that we are actually at the rest. We can look at this pattern of connectivity between the brain and the spine. So the next thing, for example, also we did in this study, this is again during the resting state. Now we can look at the ventral horn connectivity, all the areas that they are correlated with the ventral horn activity or with the dorsal horn activity. And not surprisingly, we can see that thalamus was mainly correlated with the dorsal horn. On the other hand, the frontal motor areas, SMA, M1, premotor cortex, they were mainly correlated with the ventral horn. So of course, there is a spectrum. So these areas are both correlated with the sensory and motor. You cannot separate the sensory system or motor system. But you can see that there is a tendency, for example, for dorsal horn, it was mainly correlated with the thalamus. On the other hand, for the ventral frontal lobe, the motor cortex, it was mainly correlated with the ventral horn. And we had this distribution from the frontal to the thalamus, that was areas that were correlated with those. Okay. So at UF, so this is again the work that I did during my postdoc. At UF, I've been involved in kind of trying to standardize this tool, this sequence, and make it more for the clinical use. This sequence that I showed you, it required some special adjustment that it took about 30 minutes when we put the subject in. So it cannot be used in a stroke patient. You cannot just ask them to lie in the scanner for 30 minutes while we kind of take care of the shimmy, this adjustment of the parameter to get this spinal cord signal. So at UF, I've been involved in development of the tool that we can apply to the clinical populations. For example, I'm interested in stroke patients, and using a very standard toolbox now. Like, for example, this is the multi-band sequence for those of you who are familiar with the fMRI. So there is no need for this kind of pre-adjustment before. We can acquire these very nice images of both the brain and the spinal cord. And I have developed in my lab the toolbox, all the toolbox necessary for the calculation of the connectivity between the brain and the spinal cord, and for the pre-processing of the spinal cord. So this toolbox is now freely available. I have collaborators in the different universities now reaching out, starting to use this tool and kind of replicating our results. So this is just to give you an example of what we can achieve with this more kind of the state-of-the-art protocol and sequences. So this is the activation again in healthy controls during the motor task. In this case, this was a force grip, hand grip task, using, for example, the right hand. As you can see the activation in the brain, in the contralateral areas of the cortex, contralateral sensory motor cortex, in the ipsilateral cerebellum, mainly in the contralateral thalamus and striatum, as expected. But also we can see activation very nicely in the ipsilateral side, in this case, the right side. So this is the right left reverse, the image, in the ventral horn. So this protocol, our current protocol, gives us a very good resolution and access, trying to distinguish between this ventral and the dorsal horn. So we can see that there is this distribution of activation. Now our scan is more sensitive. We can see activation around the C6, C7 level. Not only that, the other thing is, this is kind of an additional benefit that we get from this sequence, is because of this increased resolution, it's 1.6 millimeter in plane. We can also now look at the activation in the brain stem. So far, most of brain imaging studies, they have kind of uh, ignored the activity in the brainstem because we didn't have enough resolution to look at during the brain imaging to look at the activation, for example, in the reticular formation. But again, using this sequence, we can identify robustly, for example, activation in the pontine reticular formation, medullary reticular formation at different parts of the brainstem, which we know that they should be active, but again, because of the resolution issues, so far the imaging, neuroimaging studies in human, we're not able to find this functional activation at the level of the brainstem. Okay, so we have now these two. The next part of my talk and the next part that we ask in the lab is that we need to first characterize these descending pathways. For example, the reticulospinal pathway and this connection between the brainstem and the spinal cord in healthy adults, and to see what is the kind of the healthy pattern that we would expect, and then we can move on to the aging population as well as to the stroke population to study the changes. And just to give you some kind of background on the other kind of interest that we have in our lab and why we can get this information, is that mainly it's assumed that the hand motor control is governed by the cortical activation. This is what I teach in my movement neuroscience class. And balance, locomotion, posture control is controlled by the brainstem regions, which turns out not to be entirely true. So recently there are work from the animal models, from the rodents, from the monkeys studies, that they are showing that there are specialized neurons in the reticular formation that are involved in hand motor control. So this kind of dichotomy of like dividing the cortex is important for hand motor control, brainstem for more kind of locomotion or like more reflexive type of involuntary movement, doesn't seem to be quite correct. And not only in the brainstem, now from electrophysiology work, again in animal and some electrophysiology in human, we know that there are these neurons, for example, in the upper levels of the spinal cord at the C3, C4, that they send projections to the C7, C8 for control of the hand muscle. So this pathway is called purpose-spinal pathway. I will give you more information in another slides in this presentation. So we know that there are these intersegmental between, for example, the upper level, the lower levels of the cervical cord, as well as these centers in the brainstem, that they are involved in hand motor control. But again, mainly this has been shown in animal studies, this work. So using this technique, we first ask the question, can we find this activation and this involvement of these brainstem spinal cord circuits in humans during hand motor control? And again, the work is trying to characterize the pattern in healthy controls during the motor task. And this is just, again, a very brief background on the animal studies that has been done. This is mainly by the Arbers group. They have been publishing beautiful work in mouse that they are showing that in the reticular formation, there are at least two centers, one in the rostral part of the medulla, one in the caudal part of the medulla. So there is this center called lateral rostral medulla, LAT-RM, lateral rostral medulla, that has a specialized neuron that are important for different type of tasks in mouse. So when they activate these neurons by, for example, optostimulation, or they record these neurons using calcium imaging electrophysiology during different sorts of tasks, for example, during the reach, grasp task, they can see that there are a specific population that are activated. And there is this LAT-RM that's kind of sits on top of the caudal medulla, the rostral part, and it sends projections. Caudal medulla, and then the caudal medulla sends usually projection to the spinal cord. So there is this hierarchy in the brainstem from the LAT-RM to the caudal medulla, which is the most direct pathway to the spinal cord. And there are also some organization within these two regions. For example, dorsal part is more related to kind of the grooming and the hand-to-mouse kind of task. The more ventral part of these neurons are more related to the tapping, reaching task in the mouse. So there are these functional specialization within these areas of the brainstem reticular formation. Okay, so this work is kind of revolutionary. It's showing that now there are these neurons in the brainstem, not in M1, that are involved in these all sorts of hand function in the four-paw control in mouse. So first of all, we don't know yet how these results will pan out to the human. This is again another species. It might be, and the brainstem in mouse is way bigger than our brainstem, so it might be that it doesn't necessarily quite translate to humans, these findings. So first of all, we wanted to check this finding. So they mainly used electrophysiology, calcium imaging. We wanted to see, can we replicate the result using fMRI in mouse? So one of my PhD students here at UF, he built this MR compatible setup took him 2 years. And we recorded the data during fMRI while the mouse performed this kind of tapping task. So this is a force sensors. It's the water restricted mouse whenever they do it in the ITI of every 12 seconds. So as you can see, when they press on this, at this point, they will receive some water reward and they will leak it from them. So this setup, this is a head fixed mouse. So we can basically image this mouse in the scanner. This is in the high field 11 Tesla scanner at UF at Ambris. And we can look at the brain activation. So this is again the first study that has ever been done in mouse doing a motor task inside the fMRI scanner. So we can see this wide brain activation pattern in the sensory motor areas, contralateral part mainly, as well as in the striatum, cerebellum, and different parts related to the amount of force press that they made, because we can measure the amount of force and we can correlate it with the brain activity. Also, for example, with the water delivery. Okay. And next was that, as I mentioned, of course, the sensory motor areas are highly active. The striatal areas, thalamic areas are highly active. As I mentioned, we were interested in this lat RM and in this caudal medulla. And so we defined these regions of interest there. And not surprisingly, we can also get activation. So the next question was that, what is the pattern of connectivity? Can we find similar pattern of connectivity similar to those calcium imaging studies? And the short answer is yes. So this is just showing for the caudal medulla. So as I mentioned, there are two main centers, these lat RM and the caudal medulla from these electrophysiology studies. In our fMRI studies, we also see this gradient of connectivity to different parts of the cortex, to primary, secondary motor cortex, anterior cingulate cortex, and the somatosensory cortex. As you can see, the caudal medulla has the highest connectivity to these cortical areas, followed by this lat RM, followed by this kind of the medial part of the rostral medulla that we know that is not related too much, to less related to this hand motor control. So we also have this gradient across these areas of the cortex, consistent with the electrophysiology studies. The next, we wanted to look at this dorsal versus ventral kind of distinction. And so we defined again different regions of interest in the dorsal part, the ventral part of the lat RM or the caudal medulla. And our hypothesis was that the ventral, because this is a reaching and tapping task, the ventral part of the lat RM or caudal medulla should be more connected based on, again, the result of this calcium imaging study. And this is exactly what we observed. So this is the dorsal, this is the medial part, this is the ventral part of this lat RM or the caudal medulla, shown in the red and blue. And with all of these cortical region, as you can see, there is this gradient. So the ventral part is more connected to these cortical areas, as we expect. Okay, so in mouse, we showed that using fMRI, we can replicate these new results, showing these new centers in the lat RM and the caudal medulla during the motor task in mouse. Of course, the next question then, can we find these results in humans as well? So, and we used our recently developed sequences, looking at the activation of brain spinal cord during these motor tasks. And again, this is just showing similar task activation during motor tasks in the contralateral areas, cerebellum, in the striatum and thalamus. And not surprisingly, again, in the spinal cord, we can see bilateral, in this case, mostly in the ipsilateral side, again, at around C7, C8 activation. Also, with this type of task that we use, we can also see some activation at the level of the C3, C4 level in the cervical cord that we think that it might be related to this propriospinal pathway, this intersegmental pathway between different levels of the spinal cord. And then again, we perform connectivity analysis between the different parts of the brainstem and the cortex or the spinal cord. In this case, as you can see, we can find significant connectivity with different nuclei in the brainstem and with the ventral horn or with the dorsal horn of the spinal cord. Again, this is the only method that I know currently that we can do this. We can characterize in vivo this connectivity between the brainstem and the spinal cord areas. And so this is our main hypothesis, that we wanted to look at the activation in these caudal medulla. In human, we call it inferior medullary reticular formation, and in this rostral medulla, superior reticular formation. So these are the equivalent of the caudal medulla and lat RF in human, these two, SRF and IMRF. And what we see is that, again, there is significant connectivity with the brain areas, with the cortical and subcortical areas. These are the areas that were significantly correlated with these two. And consistent with the mouse data, the caudal part, which is IMRF, was more connected to these cortical, subcortical areas compared to the more rostral part. So again, so this figure is pretty much consistent with our mouse result. So it seems that we have, first of all, significant connectivity during these tasks. So these are all task-based functional connectivity, and they are also consistent with our mouse results. And then further on, we looked at the connectivity related to these intersegmental pathways in the spinal cord. So as I mentioned, there is this pathway in the spinal cord, upper levels of the cervical cord, C3, C4, that they also send projections for the control of the hand muscle to the around the C7, C6 to T1 level. And based on, again, animal models, we know that these centers, C3, C4 levels, they receive mainly information from the brainstem areas and also from the motor cortex. So this pathway is called this propriospinal neurons, C3, C4 level. And we know that this is important from human electrophysiology work. We know that this is important for hand motor control, for grasping, for reaching tasks as well. So as I mentioned here, because we have the activation and we have the signal from all levels of the cervical cord, we can look at the connectivity between these regions, and we can also look at the connectivity between C3, C4 and different brain regions. And interestingly, we also find that these C3, C4 neurons located in the ventral horn or the dorsal horn are connected also to the brain during the motor task. So to the contralateral sensory motor cortex, to M1 premotor cortex, also to the cerebellum, both ipsilateral, contralateral cerebellum. And the ventral horn in the C3, C4 was also heavily connected to the different brainstem nuclei, not the dorsal part of the C3, C4, but the ventral part, as we expect again from animal electrophysiology, that they were mainly connected to this MRF, this medullary reticular formation, which we know is one of the main, connection to the C3, C4, but also to the cerebellum. So this figure kind of shows a kind of a summary of the results that we find, that for the first time, again, we are characterizing the pattern of connectivity related to the C3, C4 pathway in humans, that the ventral horn was receiving this significantly correlated during the task with the different brainstem nuclei and with the sensory motor cortex. The dorsal part, the dorsal horn in the C3, C4 was mainly connected to the cerebellum, but not the brainstem, and also the sensory motor cortex. So this shows this kind of the pattern that is kind of consistent with the animal models. Of course, there are some differences, but to be tested using in future studies between the human and animals. Okay, so here we established that we can measure this connectivity between the reticular formation, between the cortex, between the reticular formation, between the spinal cord during the task. We also can, using our task, these force grip hand tasks, we can look at these spinal pathway, how they are connected to the cortex and to the spinal cord. And then our next question was to apply these two basically to the stroke patient and in relationship to the spasticity to see in spastic patients, can we see a difference? So this is the main topic of my NIH grant also, and the one that also I received the seed funding from the Brain Center. Is we have a three group of subject. One, we have a stroke, chronic stroke patients with a spasticity. We have a stroke patient without a spasticity, and we have age-matched elderly adults. And they come to the scanner, they perform this motor sensor motor task. In this case, I'm showing you the result of a motor task while they do this force hand grip in the scanner. And we look at the brain, spinal cord activation and connectivity, and the differences between the spastic group and the non-spastic group in this case. So in this, in my study, we also measure the H reflexes using the stimulation of the median nerve and looking at the FCR EMG activity. We look at this post-activation depression mechanism that we know it should correlate with the spasticity. We also have a Bidex robot, and we not only measure the clinical mass score, which shows kind of the resistance, but we kind of quantitatively measure the amount of force resistance using this robot. So the robot passively moves the wrist in this case at different velocity, and we look at how this velocity-dependent force resistance is related to the spastic symptom. So we can measure force, we can measure EMG during these passive movements at different velocities. So we have a very good quantitative way to measure the hypertonia, basically. And the goal is when we have enough number of patients, we then kind of correlate this hyperreflexia measure from H reflex and hypertonia measure from this force resistance with this imaging measure. So far, currently, I'm showing you only the result of 4 patients in each group, 4 patients in the spastic, 4 in the non-spastic, and 4 healthy control. So this is, of course, should be this preliminary result. It might change by addition. So we are trying to get 20 to 30 subjects per group. So we have still a long road ahead. But so far, the results are very promising. So not unexpected. We see that in the healthy control, we see activation of the contralateral side. In the stroke patient, we see mostly in the ipsilateral side because the contralateral side, there is a lesion there. So this is consistent with the previous literature as well, that the ipsilateral contralesional side of the brain, it kind of, it's overactive during a motor task in the stroke patient. And interestingly, we can see that in the stroke patient with a spasticity, there is an increased activation in this pontine medullary reticular formation. So this is related again to the hypothesis that we wanted to test compared to the non-spastic patient. Again, I don't do any statistical tests yet because we don't have enough number of patients. This is just preliminary result. Also at the spinal cord, for this spastic patient, we see this increased activation in the spinal cord compared to the non-spastic patient in the ipsilateral side. Not only that, but also at the C3, C4 level, so spinal level. So it seems that the C3, C4 is kind of related with the this increased activation in the brainstem in this patient. Again, this is consistent with kind of the hypothesis that was derived from the animal model, and we currently see similar pattern of result. We can also look at the connectivity pattern now between the reticular formation and the spinal cord, so related to this reticulospinal pathway. And what we see here is that on the ipsilateral side of the reticulospinal tract, the connectivity is increased with the spinal cord in the spastic patient compared to the non-spastic. But on the contralateral reticulospinal pathway, it's decreasing the spastic compared to the non-spastic patient. And the corticospinal tract, this connectivity in general doesn't change between the spastic and non-spastic, but it's lower than the healthy control. So to summarize, corticospinal tract connectivity decrease in the stroke patient, no matter spastic, non-spastic, and the ipsilateral reticulospinal tract connectivity seems to be increasing compared to both the healthy control and the non-spastic. So this is a little bit different from what is suggested in the literature. They kind of think that this balance of the excitatory inhibitor arises from different levels of the reticulospinal pathway. What we are currently observing is that it's related to the laterality of these centers in the brainstem, which is again consistent with some animal model. I'm not showing this here. And we are also working on the animal models, mouse models of a stroke. So here in human, we can only do correlational work. We cannot activate or deactivate these centers in the reticular formation to see how their effect is on the spasticity, but in mouse we can. So at the same time, we are also developing new models in the mouse. So we are doing these lesions of premotor cortex, basically based on some study that another group, the Japanese group did. We are looking at the H reflex. We also have basically another measure, not just the H reflex. So this is the data from my lab. We also have these miniature robot, which is very similar to this Bidex robot that we do in humans. So basically, this robot will move the wrist or the elbow of the mouse at different velocity, and we can look at their resistance. So we have these quantitative ways to look at this hypertonicity, and then this we have this H reflex. We do it both in the upper and the lower limb in this mouse while they're anesthetized. And currently, the data is promising. I'm not showing that data here. We are showing that with this model of a stroke that we are inducing that contain both M1 and the premotor cortex, we can actually induce a spasticity in mouse. Again, there are very few studies showing a spasticity in mouse. So this is one of the areas that we need to further develop, trying to further to validate our finding. And then once this model is established, then we can try to look at the activation in the reticulospinal tract. We can modulate it using neurostimulation. That's the goal of my lab, trying to see if they can improve the spastic symptoms in the mouse based on our findings. And so in conclusion, just to wrap up, I'll show you that using this simultaneous spinal cord brain fMRI provides a powerful approach to look at the function of the ascending descending pathway in humans. This is the only approach that currently I know that exists there. We can do that. And of course, it can be applied to other diseases as well that we know that it may also involve spinal cord and brain circuit together. Most of the movement disorders, as you may know. And we demonstrated the involvement of brainstem intersegmental centers. They are both active, connected in both human and mice. The results kind of follow the same pattern. We look at these connections between the medullomotor cortex. It's organized in orderly fashion in the rostrocaudal and the dorsomedial ventral axis. There is this orderly organization that seems to be preserved across the species. And we see, based on our preliminary finding, we see this increased ipsilateral reticulospinal connectivity and the protospinal pathway at the C3C4 level that seems to be related to the spastic compared to the non-spastic patient. Of course, future studies using animal models are required to causally test if the role of these brainstem and descending input in a spasticity model. And I'd like to thank again all of our funding, the Brain Center specifically for providing the seed finding and to trying to get this follow-up funding from NIH and the Florida Department of Health, and all of my collaborators and the students who made this data possible and this work possible. And thank you very much for your attention.
So Shab, thank you. That was a sort of a masterful kind of summary of putting everything together and the work that your lab has done. And it's interesting just looking at this from a clinical context where we don't always think about the underlying pathophysiology that's there. I'm just going to open it up and see if those folks who joined us have any questions that they'd like to ask.
I don't have a question, but I think that was a great presentation and just really impressive work. Very nice.
Yeah, thank you very much, David. I mean, I also thanks all of your support. So David is also one of the collaborators on this project. He provided very insightful discussions about the work. So yeah, it's an ongoing project. So hopefully we will get more patients in the next few years, and
Mhm.
it will we will have some definite answers about these underlying pathways.
Yeah, it's exciting.
So, I mean, one of the most common treatments that's done, as you know, is just giving Botox to patients to try and help with the comfort to get them more comfortable. But that's sort of, based on kind of what you're learning or discovering, that's more of a end stage type of thing as opposed to making some changes kind of more at the neural level, circuitry level. And so just getting your thoughts on, is there a chance for that kind of feedback to kind of change this plasticity at the more central level? Just want to sort of get more of your thoughts on that.
Yeah, absolutely. So this is the main work in my animal side. So basically, we have this treatment using stimulation. We use optostimulation, but it doesn't need to be optogenetic stimulation in human. We do DBS mainly. But the point is we do this repeated stimulation after the stroke from, like, for example, for 10 days from day 3 to day 15. And we can modulate. We will basically, by repeated stimulation, 3 minutes a day is not a huge stimulation either. We can kind of induce LTP or LTP depending on the parameters of the stimulation in different centers. And our goal is, if, for example, if you understand that the ipsilateral reticulospinal tract is related to the spasticity, and we know that this is a kind of delayed response pattern that develops over a month after the stroke. So the goal is hopefully to try this in the preclinical population in mouse, if we can see, and then eventually translate it to human using DBS or maybe other source of stimulation, trying to inhibit or excite, depending again with the correct type of parameters. But that's the main goal, basically, trying to go to that direction, hopefully.
And that would certainly, looking at the translation, that would certainly broaden and expand some of the treatment options that are available.
So that's the main goal. Hopefully we can do, and as you mentioned, there is most of the treatments are like mostly alleviating the symptoms. There is no, but again, the main point is that we don't currently quite understand how the spasticity is generated. So that's the first building block here toward this understanding. And if we understand and if all of the measures correlate well and we have a good hypothesis, then we can move on to the next phase of a study, hopefully toward development of new neuro treatments.
Fantastic. Let me make sure that anyone who's on, if they wanted to ask a question, they have that opportunity.
Hi, Shab. Just wanted to
I just
comment
want to say
very much for your presentation. Sorry. Looking forward to talking more because I see a lot of opportunities for collaboration, especially on the preclinical side.
It's absolutely very, very exciting. Thank you very much.
Just was to say thank you for the presentation. It was very interesting. My question was already asked, so that worked out perfect.
Perfect.
Okay. Fantastic. So, I mean, Shab, this really, we call it brain building, and kind of the way you have done this and kind of built more upon kind of the seed grant and neural injury grant. I mean, it really is kind of a living example of kind of the brain building, and certainly getting Joe excited with kind of more and more collaborations and kind of seeing the partners who are here with you. I mean, that's kind of what we're about when we sort of think about when we're trying to do brain building. So thank you very much for getting this seminar back on track. It was great work.
Yeah, thank you very much for coming also and your attention as well. Yeah, it was my pleasure, and yeah, looking forward to more collaboration across the Brain Institute as well. Yeah. Have a great day. Thank you.
Thank you.
Yeah, bye.